Olá, sejam todos bem-vindos, espero que todos estejam bem. Eu me chamo Divino, sou coordenador científico do núcleo Ganho de Estudos, do núcleo de estudo Ganho Genético, e agradeço a toda a presença de todos vocês, em especial a Rosimeire, João Edras, Catiane, que já mandou um boa tarde lá no chat do YouTube. Bom, a dica que eu dou para aqueles que estão chegando é já pegar aquele café, a água, a pipoca, um snack, porque eu acho que o assunto hoje é espetacular. Bom, lembrando que aqueles que não se inscreveram no canal, se inscreva abaixo, dá aquele like, aquela curtida. Se gostou, compartilhe também os conteúdos do canal. E antes de iniciarmos a live, gostaria de lembrar que no dia 28 de outubro, o Ganho Genético ele está com iniciativa de sorteio de livros, que será o livro sorteado dessa vez será o Melhoramento de Milho, efetuado pelo professor Rodrigo de Lima e a Luiz Borém, no caso da edição, e colaboradores. E se você ainda não está participando, vai no Instagram, olha a publicação e segue todos os passos que para participar de todas as regras para participar do sorteio. Gente, eu fiquei até nervoso porque eu recebi um boa tarde agora da minha mãe, como bom filho eu tenho que falar também. Boa tarde, saudades, né? E voltando então, o Ganho Genético tem outra novidade para vocês. Nosso próximo evento já tem hora e dia marcado, que será no dia 4/11, às 7 horas, uma live super especial, que contaremos com o coordenador do programa de Genética Pós-Graduação da UENF, o professor Dr. Alexandre Pilviana, que ele vem compartilhar um pouco sobre a experiência à frente de um programa de melhoramento de fruteiras, pontuando os desafios enfrentados no cenário de pandemia mundial, além da atuação como presidente e fundador da Rio Norte Sementes, a empresa que produz e comercializa variedades desenvolvidas na nossa universidade, o UENF, e além de abordar pontos relevantes do nosso programa. Então, se já tem curiosidade, já salva essa data, esse horário e vem participar conosco. E lembrando também que as perguntas e questionários realizados nessa live podem ser enviados pelo chat do YouTube, que serão repassados ao palestrante. Bom, dando continuidade, então, ao nosso evento, com Avanços Tecnológicos, melhoramento de plantas, hoje receberemos o Dr. Bernardo. E para levar um pouco mais esse evento, contaremos com a mediação do professor Dr. Messias Gonzaga Pereira. O professor Dr. Messias Gonzaga Pereira ele possui graduação em agronomia, mestrado em genética e melhoramento de plantas pela Universidade de Viçosa, doutorado em plant breeding pela Iowa State University e atualmente é professor titular da Universidade Estadual do Norte Fluminense. Além do mais, ele possui grande experiência na área de milho, mamão, e maracujá também, com diversos cultivares lançadas, pesquisador de produtividade A1 do CNPq e cientista do nosso estado pela FAPERJ. Então, convido a gente se juntar-se a nós, o professor Dr. Messias. Olá, professor, boa tarde.
Boa tarde, Divino. Muito obrigado aí pela oportunidade de participar com vocês. Eu queria, em seu nome, parabenizar todo o núcleo Ganho Genético pelo trabalho que estão fazendo, pela divulgação de temas tão importantes. Estou à disposição, então, para moderar essa live tão importante. Acredito que podemos já fazer a apresentação do nosso palestrante de hoje.
Perfeito,
que é uma honra para nós. Se quiser trazer o Dr. Bernardo. Today, we have the honor to receive Dr. Bernard. Nice to meet you here. It's a pleasure. Dr. Bernard, I will read a little bit about his curriculum. Bernard is professor and endowed chair in maize breeding and genetics at the University of Minnesota, USA. He obtained his undergraduate degree in Philippines in 1984, and his PhD in plant breeding from the University of Illinois in 1988. Dr. Bernard developed the widely used G-blood procedure in 1994, and most of his work has focused on marker-assisted breeding. Dr. Bernardo is director of the Plant Breeding Center at Minnesota. has written 2 textbooks, and teaches graduate courses in plant breeding and in professional skills for scientists. Today, we have the honor to hear his talk about the title of his presentation is Maize Breeding, Past, Present, and Future. So, please take your time, and again, thank you very much.
Well, thank you very much. Thank you very much for having me. I'm assuming you can hear me well. If you cannot hear me, just let me know. It is a pleasure to speak with you today online in this platform. It's a pleasure for me to talk about the history and the present status and perhaps speculate on some future developments in maize breeding. And so, a bit of my background, I finished my graduate degree at the University of Illinois, and that's where I first learned about maize breeding. After graduation, I started working with a seed company in Illinois, and I worked with a seed company, a maize seed company, for 9 years. And that's where I learned a lot about maize breeding and how maize breeding is done in the private sector. A lot has changed since that time. And in the past 24 years, I've been now working in universities, first at Purdue and now at the University of Minnesota as a professor in maize breeding and genetics. So what I'd like to do today is again talk about maize breeding and where we're headed, where we have been and where we are and where we are headed. And I'm not going to use any slides to the extent possible. When I give talks these days, I don't use slides anymore. And so if we think about plant breeding and what are the necessary requirements for releasing a good variety of any crop species, I think there are 3 ingredients or 3 factors that are needed to release a good variety. Number one is we need germplasm. Without germplasm, without good germplasm, we cannot release a variety. Number two, we need methods and tools and resources. Without methods, tools, resources, including funding, then we cannot do the work that is needed to release a variety. And third, we need people. We need people to do the work of breeding in any species. So we need germplasm, we need tools, methods, resources, and we need people. And so what I'd like to do is talk about the history of plant breeding on the basis of these 3 factors: germplasm, methodologies, and resources, as well as people. Now, we know, and I'll be talking about it mainly from a perspective of maize breeding in the US, knowing that developments in the US have then spread across the world, maize breeding around the world, in Asia, in Africa, in South America, and in Europe. And so, when we think of the development of germplasm in maize breeding, back in the before the 1900s, we of course know that open pollinated maize varieties were grown in the US and in around the world. So with open pollinated maize varieties, these were segregating populations that the producers can save seeds and plant the seeds for next year's season. Now, George Harrison Shull, in the early 1900s, then published a paper entitled The Composition of a Field of Maize, in which he noted that basically when you have a population growing in a maize field, it is a mixture of heterozygotes, and that he proposed what maize breeders should do is, instead of growing open pollinated varieties, that we should strive then to reproduce, to first find a good hybrid and be able to reproduce that good hybrid through inbred parents. At that time, uh, scientists thought that there might be some practical difficulties with Shull's procedure of the inbred and hybrid system for developing maize varieties. And so that's why in the early 1910s, maize breeders figured out that instead of growing single crosses, which is a cross between 2 parents, they first grew double crosses. So instead of the cross A by B, they would have a cross between four parents, A by B, and then C by D, and you cross these two F1s to get your double cross. And the reason for that then is to have good seed production for the seeds that the farmers would grow. And so we saw then the development of different germplasm groups. Breeders started now self-pollinating the open pollinated varieties that were grown at that time, and started then developing inbred lines of maize. And what happened then is that there were different key, or very important, there were the key lines that were found to do well, and that led to newer and better versions of such lines. In other words, in maize breeding, there were different pedigrees that were established. And so you have pedigrees such as a B73 pedigree, pedigrees such as B37, B14, Missouri 17, WF9, and so forth. So instead of having this diverse germplasm, open pollinated germplasm, the germplasm of maize then became more and more elite, but became more and more narrow. And that's where we are today. Most of the germplasm now is developed not by universities, but by different seed companies. And now we have groupings of germplasm in maize into different heterotic groups. But what has happened is that the maize germplasm base, again, has become a lot narrower these days. And breeders have begun to ask, well, how long can we keep doing what we're doing? How long can we keep on making better and better versions of the same basic genetic background, or the same basic groupings of such lines? So I think now, as we look into the future, it will become increasingly important to find new sources of germplasm. And it becomes a challenge because the new sources of germplasm are typically not as high performing, or perhaps not as well adapted as the most elite germplasm that we have. And it is tempting to focus on the short-term genetic gain and just look at high-performing hybrids for the here and now. But I think any maize breeding organization needs to look at the long term. If you think about it, even though we are concerned about genetic diversity, a goal of a breeder is to exhaust, or to use up, all the genetic diversity in a given cross. By that I mean, if you cross parent 1 by parent 2, and you get all of the good genes from parent 1 and all of the good genes from parent 2 somehow, and you combine that into a new line, say line number 3, then you have exhausted the genetic diversity, because you had two lines and now you have one line with all the good genes. But you have exhausted the genetic diversity present in those two, and that is by design. You should be happy if you're able to do that. But what is needed then to sustain the long-term progress is that as maize breeders are developing newer and better hybrids, there has to be a flow of new germplasm at the start. So you have good improved germplasm going, or being developed on the one end, but you need a steady flow of new germplasm that goes at the very beginning for sustained improvements. And you need to ask, well, where will this germplasm come from? I think a key source of such germplasm is germplasm that is not necessarily elite in your own geographic area, but is elite elsewhere. And we've already seen that major companies have become successful in using germplasm that is elite in other places around the world, and adapting it and bringing it to the home environment, and using that in breeding. Or just to make the example, just to give an example, breeders in the US have successfully used germplasm, say from Brazil, germplasm from Argentina, germplasm from Thailand, and other sources, meaning elite hybrids in Brazil and Argentina and Thailand and other countries, brought them into the US and used that in the breeding process to expand the breeding pool. I think that as we look forward, it is actually the germplasm that will differentiate different breeding organizations. Meaning that if you look at the different breeding companies, they will probably be using pretty much the same breeding tools, the same breeding methodologies. They will all have good people who are devoting themselves in the breeding effort. And what would differentiate one breeding organization from another breeding organization is then the germplasm base they have. Because again, you need all three ingredients: germplasm, people, and methods and resources. Now, if we look at then the different methodologies that have been developed through the years, it's fascinating. It really is fascinating to look at the different methods that have been developed. And personally, I take great satisfaction in having been part of about 25 or 30 years of seeing those developments of methodology in maize breeding. If you look at in the past, selection used to be simply phenotypic selection, meaning that you would either evaluate individual plants or lines out in the field, look at them visually, take measurements. You would make hybrids or test crosses of different maize lines, and you would evaluate them phenotypically in the field for traits such as yield, grain moisture, plant height, stalk lodging, root lodging, flowering date, disease resistance, and other traits of interest. And in fact, I say when I started my career and I started my studies as a student of plant breeding back in the mid 80s, plant breeding was quite simple. It was quite easy. You crossed good by good, you developed progeny, you evaluated them in the field, you identified the best, and then you repeated that process of crossing good by good, evaluate in the field, select the best, and repeat that process again. Now, my life as a scientist became a bit more interesting and perhaps a bit more complicated when I had a fellow graduate student at that time walk into my office and tell us about this new wonderful tool called restriction fragment length polymorphisms, or RFLPs. This was about 1986, if I recall correctly. And so we had to learn about RFLPs. That was the first type of molecular marker that became used routinely in maize breeding. And so molecular markers really were not new. Even back in the 1970s, there were protein markers called isozymes that have been developed. The issue is that the isozymes, there weren't too many of them. You might get 10 isozymes, 12 isozymes, maybe 20 or so if you're lucky. But now with RFLPs, breeders had at their disposal a large, potentially large number of molecular markers that were distributed throughout the genome and that were that were neutral by themselves, but they could be linked to different loci of interest that would control the different traits of interest, and hence the era of QTL mapping. Now, when it started out, molecular markers were fairly expensive. I remember the first time that I got exposed to marker work was when my professor at the University of Illinois called our entire group and told us we should meet in the field because we will be collecting leaf samples for DNA analysis. So we drove out to the field. We were given gloves. We were told to wear gloves so that our DNA from our hands would not potentially contaminate the corn DNA. And then we were given a pair of scissors, and then we were asked to get leaf samples for DNA analysis. We were asked to cut leaves that were about 6 inches to a foot long. So we cut the leaves from these individual plants. We put them in a bag. We put them in a cooler with some dry ice, and the cooler was driven to the local airport. There was an airplane waiting at the local airport to take these precious leaf samples to a lab in Iowa. And I told myself, this is a very expensive operation if this is what we need. And at that time, it was indeed expensive. The price per data point, meaning that one RFLP marker in one sample, was probably in the $5 to $10 range. But my advisor told me that, Rex, you need to think of applications of DNA markers when they are quite cheap. In fact, when they are as cheap as a paper clip that we use to put up the bag when we pollinate corn. And at that time, I thought that would never happen. But sure enough, here we are today, where the cost per data point of a single nucleotide polymorphism or SNP marker is about the cost of a paperclip. In fact, even cheaper. And this cost differential then has driven how we do maize breeding today. And we can look back at the history of maize breeding, and we can see how different methodologies were developed for finding QTL, quantitative trait loci, or marker trait associations, that can be done by linkage mapping in a cross between two inbred parents. It can be done in an association mapping panel, for example. And beyond that, after that, another key methodology that was developed, not just in maize, but maize was a main species for plants, was genome-wide prediction or genomic prediction or genomic selection. And now, right now, genomic selection or genome-wide prediction is routinely used in different maize breeding organizations, maize breeding companies in the US and around the world. And what has happened is that the cost of genotyping in maize in the US is quite cheaper than the cost of phenotyping. For example, the cost of genotyping now for several hundred to a few thousand markers is about 10 US dollars per sample. The cost of one plot for field evaluation of corn in a yield trial is about $15 per sample. And if you say that in first year yield trials you need 6 locations, that means for phenotyping you need $90 per individual or per hybrid, and for genotyping you need $10. So that 1:9 ratio or cost differential then has driven the way that we use markers, and a lot of selections now in maize breeding are made based on molecular marker data. Now, that is just one tool that has been developed in maize breeding, and we can point to many other tools that have been developed. One important tool is the use of doubled haploids. That it used to be that when I started my career as a maize breeder, the path was clear. You cross two parents, you got the F1, you self the F1 to get segregating progeny in the F2, F3, F4, F5, F6, F7. And so you needed 6 to 7 to 8 generations to come up with homozygous lines. However, with the development of doubled haploid methodology, now it takes only 2 generations to develop a doubled haploid line that is completely homozygous. And that has radically transformed how maize breeding is done, because you could start from 2 homozygous parents to a homozygous line very, very quickly, so that the focus now has been on testing the performance of these homozygous lines. You know, I call these as power tools. Just a side note, the power tools in corn breeding. You think of the tools we would have around the house. We could have a hammer or a saw. So for a saw, for example, to cut wood, well, yes, you could use a saw, but it is quite slower and less precise than if you use an electric saw or power saw. It's the same thing. We have these tools such as molecular markers and doubled haploids that speed up the breeding effort. Now, those aren't the only tools, and we can point to quite a few other tools. One is the combine, the plot combine. It used to be that when I started my career as a plant breeding student at Illinois, my professors somehow still harvested some of their yield trials by hand. Didn't understand why, but imagine harvesting a yield trial by hand and having to shell that corn. And I know that's still done in many parts of the world today, but now with big 4-row combines, twin plot combines, then that has increased the capacity for phenotyping in maize breeding. And finally, another tool that has become important is fast and cheap computers. One of my professors, when I was at Illinois, Professor George Sprague, a famous professor who came up with the concepts of general and specific combining ability, Professor Sprague told me that in his day, they could not have winter nurseries. And the reason they could not have winter nurseries is that they could not get the data analyzed in time to make decisions on what material to send to winter nurseries. But now we're at this time when we have fast computers and statistical packages to help us analyze and visualize and interpret our data so that we could now make breeding decisions quickly. And I mentioned another tool that we have is year-round nurseries. So if we're in the US, then we can send our maize to a nursery in Puerto Rico, for example, or in Hawaii, where we could grow up to 7 generations in 2 years. And so that speeds up the breeding process as well by having multiple generations for advancement. And we can put on other tools such as embryo rescue. By embryo rescue, we now can even shorten the breeding cycle because the seeds do not have to mature, and they also don't have to go through that germination phase. So that cuts down on time, and that cuts down on the makes breeding a lot faster. There are other tools that have come to the horizon, obviously, and these include tools in phenomics. Drones have become commonplace in maize breeding these days to make data collection faster and on a much wider scale. Now, when we look at tools in the future, this is just speculation. One tool, both in the present and in the future, is genome editing, of course, is a tool that I believe will become increasingly important. The challenge is that we need to know the types of edits, what we need to edit, and the types of edits that are needed. We need to have information on that. Another tool or methodology or approach that I think will become increasingly important, and it's something that I've started to work on, is this idea of targeted recombination. When we think of the different aspects of breeding on which we can have control, a maize breeder actually has a lot of control over the entire process. The maize breeder can control which parents to cross, how many progeny to make, which testers to use in, or which hybrids to make, how many locations to use in phenotyping. The breeder has control on how to use drones. The breeder has control on how to use molecular markers. But one aspect in which the breeder does not have control yet is recombination, or the meiotic process. Meiosis or recombination, of course, remains a random event in which the progeny that we get are simply a function of just whatever we get from a given cross. Now, targeted recombination refers to being able to induce by CRISPR or to select for recombinations in those places, exact places in the genome where the breeder thinks it would be helpful to have recombination. So what happens is that with genome-wide prediction and marker effects of the different markers on the genome, a breeder can begin to predict where recombination should occur on chromosome 1 to maximize performance, where recombination should occur on chromosome 2 to maximize performance, and so forth. And so I think that one tool that can be added in the future going forward is this whole idea of being able to exert or to have some control over recombination so that we can precisely say I want recombination here and there and other places in the genome, because that's where we think performance would increase. I think another tool that maize breeders can and should be working on is speed breeding. You could imagine, corn, it takes us about 4 months or so to grow out in the field. But what if by some means, either genetic means or by manipulating the environment, we can reduce the amount of time required by corn from seed to seed? What if we can reduce that to 3 months? What if we can reduce that to 2 months? What if somehow we can reduce that to just 1 month from seed to seed? And maybe the plant will be small. Maybe we'll just get 10 seeds per plant instead of 400 seeds per plant. But if 10 seeds is enough, we would gladly trade that reduced seed amount in the breeding process for a faster, quicker generation interval. So I think that is something that it's well worth breeders' time and attention looking at into the future. And finally, so we've talked about germplasm, I've talked about tools and methodologies. Now I'd like to talk about the people, the people aspect of breeding and how that has changed from the past to the present. It used to be that breeding was done, maize breeding was done in the universities mainly, and then more and more it shifted from the universities to breeding in the private sector in seed companies. And we saw also breeding shift from more of a single breeder operation to teams of breeders working together. What I mean is that when I started my career in the seed industry, it used to be that you had one breeder working with 2 or 3 technicians, and then maybe a secretary in one station. And this breeder then had responsibility for that breeding program in a certain geographic region. And you replicated that system. You had in another region, you had one breeder working with 2 or 3 technicians and a secretary and part-time workers to run the breeding process. And a breeder had control over his or her resources. Of course, they interacted, they exchanged, but a breeder pretty much had control over resources. Things have changed these days in the sense that now, instead of individual breeders, you have teams of breeders working together in the same station, sharing technicians, sharing combines, sharing planters, sharing resources. And for a breeder, and these breeders need to work together with a fixed amount of resources. So there's give and take, there's back and forth, there's negotiation with your colleagues to partition these resources for the different breeding efforts. We also see an expansion in the expertise of those involved in the breeding process. It used to be that breeders, when you say breeder, well, you understood that yes, you got a master's or a PhD in plant breeding and genetics, and you'd be working with people with allied expertise. You might have a pathologist working with you. You could have statisticians working with you, but the breadth of expertise in maize breeding has expanded tremendously, to the extent that if you look at breeding organizations involved in maize, there's a big emphasis, for example, now on data science, on machine learning, on artificial intelligence. And breeding organizations now are looking for diverse talent, looking at talent not necessarily in breeding. They're hiring people who have not worked in any breeding organization before, thinking that it's sometimes easier to teach a computer scientist maize breeding than it is to teach a maize breeder computer science. And so that I think that is reflected in the curriculum that we have in breeding. It is reflected in the courses that our students now take, in plant breeding students take these days. Now, in the future, I actually think that there might be fewer breeders needed in maize. A reason for that is, I believe, that many of the breeding decisions can now be automated. By that, I mean it is possible to train a computer to replicate the breeding decisions that are made by a breeder or a team of breeders. So instead of hiring additional breeders, for example, for the initial stages and initial selection decisions made in a breeding program, a computer should be able to do that, so that the breeders, the human breeders then, could spend more time with fewer material and make better breeding decisions on the germplasm or the remaining lines and hybrids that have already been pre-selected. I know it's a bit scary to think about that, but I know it's possible, and I think breeding organizations are going that way. Does that mean that a computer will replace breeders? For some aspects of the work, yes, but certainly not completely, because as a rocket scientist once said, yes, you can train a machine to perform well under conditions that are anticipated, but when you come up with unexpected situations, that's where the human intelligence and human intuition becomes extremely important. So to summarize, what I see as the past and the current status and the future of plant breeding, of maize breeding and plant breeding in general, is that maize breeding is becoming more and more like a factory process. It already is a factory process. It's not your small backyard artisan process, but it really is like a factory. If we think of a factory, you have some raw material. So your raw material could be wood or plastic or steel. This raw material comes through some manufacturing process to create something. What you've created goes through a quality control process, and after quality control, then you release this new toy or new car or new whatever it is, gadget that you've invented. And in the same way, in maize breeding, your raw material is your germplasm. It goes through a manufacturing process, which is the breeding process. It goes through a quality control process that is your field trials or nursery trials or molecular diagnostics. And finally, a product is released, and that is your improved hybrid variety of maize. There always will be some uncertainty to it because of we're dealing with a biological material. If you think of steel or wood or plastic, you can shape it and mold it exactly the way you want. In maize, there's meiosis, there's fertilization, and there's a big part of the unknown. However, I see it more and more as becoming a precision, efficient manufacturing type of process. And I think that's where we're in a way, in many ways we are, and we are headed even more so in the future. Thank you so much for letting me share my thoughts today on the past and present and future of maize breeding. If there are questions, I'd be glad to try to answer them. Thank you so much.
Okay, thank you very much, Dr. Rex Bernard, for the wonderful talk. It's quite new information and a challenge to have the whole idea. And before we get started the question, let me ask you your first question for you, okay? In this factor process that you imagine for the future, do you think that we have some space for recurrent selection or not?
That's a great question. I think is there space for recurrent selection? I think there has to be. I think there has to be space for recurrent selection, and because we know that there's a lot of power in that recombination process, and that the selfing, the inbreeding process, especially with doubled haploids, is so quick that in a way it becomes a game of chance. It's in a way, a lot of it is hit and miss, depends on large populations, depends on trying just by chance capturing what you want. Whereas in recurrent selection, yes, it does take time, but in the end, it is a slower but steadier and more predictable approach, if you would. Then, so we need both, in my opinion, and I think the most successful breeding organizations will have some form of recurrent selection. It doesn't necessarily have to be long-term recurrent selection. It can be short-term recurrent selection, such as selection for 3 or 4 cycles, and that's it, in a biparental cross. It does not have to be broad-based. It can be in a biparental cross, but it is still recurrent selection nevertheless.
Okay, thank you. Let me see if we have a question. Okay, we have our first question. The question is from Thales Oliver. Nice talk, Dr. Bernard. When it comes to plant breeding maize for the negative impacts of climate change, do you think our biggest challenge is available germplasm, or there is still a lack of technology, human resources? Okay, I guess that you can see the
Yes,
question.
yes. That's a great question. Thank you, Thales. I think it's not really a limit. I don't think it's a limitation. It's more of people or technology or germplasm, but it's more breeding for the here and now versus breeding for future environments and how quickly changes in the climate would happen. And so if you look at a maize breeding program, it would take 6, 7, 7, 8 years. And so what we really need to do is try to anticipate what the environments would look like in 8 years. And so uh, I think maize is so diverse that you could just about breed for anything. I mean, that's an overstatement, but maize is just so diverse. There's a lot of diversity that if you know what to breed for, chances are you'll be able to accomplish that goal. And so the thought then is, well, it's how serious are we in breeding for drought tolerance? How serious are we in breeding for nitrogen use efficiency? How serious are we in breeding for a type of maize that can grow quickly to provide cover to the soil? How serious are we in breeding for maize that has a good root system to hold onto the soil? And of course, there's give and take. I mean, you can't have it all, but if you change one part of the maize plant, then you could be changing other parts. So I think, yes, the challenge is there, but it's not to say that breeders have made tremendous progress actually in a way. We didn't call it climate change, but tremendous progress in breeding for example, drought tolerance. The corn today is a lot more drought tolerant than the corn from previous years. In the drought year in the US of 1988, corn did quite poorly, but in more recent droughts, you don't see as big a loss in performance anymore.
Okay, we have another question from Gian. Excellent lecture, Doctor. What do you think about the triangle genotyping, phenotyping, and environment to improve the accuracy in maize crop models?
Thank you. Thank you, Gene. So crop modeling is something that I did not mention. But yes, when we talk about looking at the response of maize to climate change, then and predicting the performance of maize in specific locations, then crop models become quite important. And yes, to do crop modeling well, you need good information on the genotype. You need good information on physiology. You need good information on the environment. And it used to be that we did not pay too much attention to collecting environmental data. But we're doing, I think, a lot better job now at collecting environmental data. The challenge would be that there are certain aspects of the environment that stay the same from year to year. Soil type, for example, stays the same from year to year. Cultural management practices stay fairly the same from year to year. But then there are other aspects such as precipitation or rainfall and temperature that may not stay the same for year to year. And so, yes, we can predict which maize hybrids are best to grow under drought or which are best to grow under well-watered conditions. But until we know whether we will have a drought or well-watered conditions in 2022 in a certain location, then we'll always have that limitation. That being said, there's quite some work now being done to do exactly that, to try to predict in a certain location what would be the temperature next year and what would be the precipitation rainfall next year. And it'll be interesting to see how well those predictions work.
Thank you. Well, we have another question. It's from Divino. In the past, the plant breeding process was based on phenotype. Today, It is based on phenotype, genotype, and association with molecular techniques and bioinformatics. In this context, in the future, the selection of superior genotype will be based on what?
I think in the future, it'll be based on all available information. It won't be just one source of information, but it'll be all available information that we have. It'll be phenotypic data, it'll be marker data, it could be multiple traits. Instead of looking at one trait at a time, for example, when we evaluate yield, we also look at moisture content and maybe even protein content and starch content, because those are correlated to yield itself. So I think it'll be looking at all available types of data and integrating these types of data in the selection process. And so what happens then is that in maize breeding, what a skill that becomes extremely important is the ability to analyze, to visualize, to interpret large amounts of data, to make selection decisions based on those large amounts of data, and to move on. You make your decision and you forget about it and you move on to the next selection decision you need to make without trying to second guess yourself whether you made the right decision or not.
Let me ask one more question, please. How do you compare the potential of in the future, the genome editing versus transgenic technique?
It's a great question. I see them as complementary. I don't think it'll be one or the other. I think it'll be both. A lot of that potential would be much of the answer to that will be driven by politics and regulation rather than the science itself, on whether there are on genome edit, whether or not in a certain country genome edited products will be subject to the same type of restrictions as transgenics. A challenge is that with genome editing, of course, we need to know what the targets are and what types of edits to make. And I think those types of targets need to be genes with large effects. And for yield in maize, there really aren't genes with large effects. If there are, they have been fixed in the germplasm we're working with because they have large effects and they're important. And so for things such as yield, what we're left with are small effect genes, and any edits that we make would have smaller effects. So gene editing will become important for traits such as disease resistance, insect resistance, flowering date, morphology, these types of traits for which there could be major genes that have larger effects.
Okay, it's kind of similar to transgenic in
Exactly
way.
Right. Yes,
okay,
exactly
okay.
Right. Yes, yes.
Okay.
Yes.
Well, I guess that we have no more question for you.
All right.
And, okay, I like to again thank you very much for your wonderful lecture. It was, let me see. We have one question again. I'd like to know what the pandemic situation can change the work with plant breeding. Okay,
Yes,
sorry.
it's a great. No, it's a great question. So with the pandemic, I think we have, for example, more online seminars such as this. It's a good thing. I think just a lot more. We're learning to how to work together without being physically present with each other. So I think at least that is some positive development that has come. So breeding is a team effort, and so I think with the pandemic, breeding teams have learned how to work efficiently together. Yeah.
Okay. Well, let me see if we have more question. Question. I guess that's it.
Okay.
So
Well,
thank you.
you very much.
I guess that it was. I'm sure that everybody appreciates so much your talk. It's quite visionary in terms of the future of plant breeding, especially maize. And for me, it was great. I started my PhD in 1990 with RFLP map, doing some TTL analysis, and it was kind of going to the past. Here, when you said the RFLP,
Yes,
it was the start of the molecular markers in terms of application, real application in plant breeding. Okay,
right.
Thank you. I guess that, sure.
Well, thank you, and have a good evening. Thank you so much. Bye-bye.
Thank you. I return to Divino. I guess that he will tell us something.
Ok. Obrigado. Todos estão me ouvindo? Sim. Obrigado a todos. Thank you, Dr. Hex Bernardo, for excellent lecture. And thanks everybody for coming and watch us. And I will speak Português now, alright? Obrigado a todos. Tenham uma boa tarde. Obrigado pela audiência, que foi bastante legal a palestra. E até a próxima, então. Gente, tchau, tchau.